My name is Gamze. I am an assistant professor of biomedical informatics and computer science at Columbia University and New York Genome Center. And today I'm going to talk about privacy enhancing, well, it says technologies, but I will only have time for one technology and an application in genomics. So before getting started, people have been doing data protection for years. It's not news, but it's a little bit new in the space that we are in. I'm not motivating the problem here because I think that by now everybody probably is convinced that the genetic data is sensitive, and whether it's ethical, legal, or institutional limitations that in certain cases we cannot share the data. So data comes in different forms. When we transit the data, we have secure protocols. When the data is at rest, the data is encrypted. But when the data is in use, it's very vulnerable. And that's the space that I'm going to talk about today is how can we analyze genetics data while protecting the privacy. So mostly talking about security. Now I'm a little worried because we just heard a talk by a real mathematician, and I'm going to be a fake mathematician and talk about cryptography. I was not formally trained in this. This is all self-taught, so bear with me and please be kind. What is cryptography and why do we care? And why do we think that this could be useful for our purposes? So it provides secure communication in the presence of malicious third party. So for example, if you want to outsource your analysis to the cloud, you may not be able to do that for every data point that you have because it depends on the consent. And for example, in Europe, because of GDPR, even the de-identified genetic data can be considered personal data because it's pseudonymized, so you might have to encrypt it. But if the data is encrypted in the cloud, then how can you actually utilize the cloud resources? So when we talk about malicious third party, it could be the cloud, or it could be someone peeking into what is happening into your shared server or the cloud. So we categorize the cryptography primitives into two categories unofficially, but this is just me and my postdoc Sengman decided that we will do it this way. Encryption, it could be an algorithm and a key to transform a plain text into a ciphertext, meaning that you're encrypting the data and you might be doing something with it. And the protocol is usually two or more parties jointly computing a function without sharing the input. So when we talk about encryption, we talk about symmetric or public key encryption, meaning that you either have one key for encrypt and decrypt, or you have two keys, one for encryption, one for decryption. Homomorphic encryption is a type of encryption that allows you to compute on data, and today's talk will be mainly focused on that. And then on the other hand, we have protocols like key exchange. How do we securely exchange our private keys, or multi-party computation, which is another widely used tool, especially in genomic space. I was thinking that I would talk about that too, but I think 30 minutes will not be enough. So next year, come back, I'll talk about multi-party computation. And then we have things like zero-knowledge proofs, which are the base for the blockchain technology, for example. So what is homomorphic encryption? As I mentioned, it's a type of encryption that allows you to compute on ciphertext. So then you can delegate the computation to an untrusted server without revealing the data. So how it works is you first encrypt the data, and then you send it to cloud, and you execute a function on the encrypted data, and then you get an encrypted results, and then you download it locally, and then by using your private key, you can decrypt the results. And ideally, that the results that you get from this computation is same as what you would get if you do all these operations in plain text. However, it's computationally expensive and supports limited operations, which I will talk about why it supports only limited operations, such as addition and multiplication. So what does homomorphic encryption is based on, and why we know that it's very difficult to decrypt, by decrypt meaning it's very difficult to basically hack into. So it is based on this problem called the ring learning with errors. So it's a mathematical problem that is believed to be so hard, even for quantum computers, And it's basically the cornerstone of modern cryptography and homomorphic encryption specifically. So what is it? So in this particular problem, we live in this polynomial ring space. And in this particular case, we talk about polynomials that have coefficients that are in integers with modulo q, meaning that if you have a coefficient that is larger than q, it wraps around because it's modular arithmetic, and then n determines the number of the maximum degree that you can have in this polynomial. So the ring learning with error problem is such that you have a secret polynomial, Sx, and then you have your Ax, and you're telling, hey, multiply my polynomial Ax with my secret Sx, but also, add some noise to it so that it's more secure, and now you get Dx. And then it's been shown that if you don't know Sx, it's very, very difficult to find out what A is, even if you know B, because of E. And because we live in the polynomial ring space, when we do this and when we encode everything with polynomials, any operations that you're going to do with A has to be just multiplications or additions. You cannot do divisions, because when you do divisions, your coefficients might go outside of the ring. So you have no guarantee that you're inside the ring. And that's why homomorphic encryption is very limited, because you cannot do divisions, you can only do multiplication. But this is a problem when we talk about classical bioinformatics algorithms. So now you have to rethink how you're going to do your algorithm based on these limitations. So let's talk about how this works. So we have the secret polynomial, Sx, which will be your secret key in encryption. And you have a message Fx that you want to encrypt. You encode your message in a polynomial. Either you can use coefficients, for example, to encode your data points. And then you define your A and B such that you can do any kind of polynomial operation on M. But if you don't know S, you can never recover M. So that's the whole idea. So that's why it is based on the ring learning with errors, and that's why you can only do the polynomial operations on it. And then the decryption is basically when you get the decryption, you will have the error on it, but error is so small that it doesn't affect your results. It's so small that it doesn't affect your results, but also it adds so much to it that even if you know S, you cannot recover M, because even if you know without S, you cannot recover M because of E. So that little error actually makes it both secure, but also when you decrypt it, it's accurate. And just to give you an example how it works, it's very simple. You can basically add two ciphertexts, two encrypted texts that are expressed as two polynomials, A1 and B1, A2 and B2. And then at the end, when you decrypt it, you will basically end up with M1 and M2. And this multiplication works in a similar way. You can just look at Wikipedia how this works. I'm not going to go through it, but basically everything is encoded with polynomials, and you can only do the operations that lives in the polynomial ring. There are different schemes, and they are going. So there's an ISO standardization. Every year there's a workshop. People get together and standardize certain things. And these schemes are all named after the initials of the last names of the authors. These are my favorite schemes, BGV, BFV, because in genetics, genomics, we live in the count space usually, and genotypes are discrete, so integers are really nice to work with. And also, you can do SIMD-like operations, so you can pack multiple messages into a single ciphertext and then execute them in parallel. So this library TFHE that does only bit-like operations is very good if you have hash tables, lookup tables. It can do it very fast, but it cannot do other operations really fast. And then if you want to work with real complex numbers, there are also schemes, CKKS, but it was known to be not as secure as the others, and there were some papers hacking into it. So, but because of all that, the computations in homomorphic encryption can be 1,000 times slower than the plain text computations. As I said, it has limited functionality. You cannot do many nonlinear things. So, for example, people are very interested in doing privacy-preserving machine learning, but it's really difficult to compute functions like value or anything nonlinear. And then as you do more operations, that E term will get increased. And at some point, the E becomes so large that you cannot decrypt anymore. When you decrypt, you get wrong results. There's a technique called bootstrapping. Basically, it's decrypting while the data is still encrypted. So you're decrypting in the encrypted space. So you're turning one encryption to another type of encryption, but it's very expensive. So we usually, to get around that, you basically design an algorithm such that you, ahead of time, you know the depth of your circuit so that you can budget for your error accordingly. So that's the gist of how homomorphic encryption works. So I just want to now talk about a little bit how we can use this to quantify gene expression from RNA-seq reads. So the scenario here that you have, let's say, thousands of individuals. You're part of a consortium. You have thousands of individuals' RNA-seq data, raw reads. You need to quantify gene expression. It's going to be too expensive to do it locally. So you want to outsource this data, but somehow there is no consent or there's a law prohibiting you to do any kind of analysis in the cloud. And the reason is that each of the next generation sequencing reads will have the bits of personal genome of the individual. So here what we want to do is we want to encrypt these reads, send it to the cloud, quantify the gene expression, and then take the encrypted results back and then decrypt it locally. So I'm sure that you guys all know, but quick overview of RNA-seq is basically what you're getting is you're isolating the RNA and then turning it into cDNA and then sequencing the DNA. There are two types of approaches how you quantify transcripts or gene expression. One is alignment-based one, and which was discussed yesterday. You know, you just have a seed sequence in the read, you map it, and then you extend until you find the correct location of the read, and then you basically aggregate all the reads underneath the genes and then count them, and that count is a proxy for the expression of that gene. And then there is the pseudo-alignment-based approaches such as Callisto that uses a graph structure, and it can also do the novel transcript discovery by doing that. So we cannot directly take these algorithms and adopt homomorphic encryption because most of the time they use conditional statements such as if, while, and these are very difficult to execute because think about an if statement. You have an encrypted data point that you are comparing to another encrypted thing. You don't know what this is. You don't know what this is. So you cannot compare them. So you need to basically turn it into either bit-like operations or express them as multiplications or additions that can live on the polynomial space. So basically the idea is that we have a client that encrypts the reads using homomorphic encryption, sends them to server, and then the server homomorphically calculates the gene expression and sends back to client. Okay, so how are we going to do that? If we are not going to use if and while, if we're not going to use existing algorithms, can we come up with a simple algorithm that uses only multiplications and summation such that we also know the depth of our circuit so that we can actually express it in the homomorphic space? So, I mean, I did not know that there were going to be so many K-mer talks yesterday, so I made the slides. But what we are going to do is basically K-mer batching. So the idea is that you divide so you create these gene sequences. For each gene, you take all the exons, you concatenate them, you create these K-mers, that is your reference K-mer indexing, and then for each gene, you count how many times you see that K-mer. And then you have the read, you do the same thing, and then now you're going to calculate the frequency of the K-mers that are at the overlap of your gene and the read. And if that number is larger than all the other genes, then that read is likely originating from that gene. So what you can do basically, you can create a vector from the gene with all the frequencies of K-mers, another vector from the read with all the frequencies of the K-mers, And then you can calculate the dot product that will tell you basically the frequency of the overlapping k-mers. And we use this as a proxy to gene expression. And this k-mer representation allows for optimizations because, so you can imagine that there are many k-mers that shows up in many, many different genes, and they are not very useful. So we do this entropy pruning. We calculate the entropy of a k-mer, meaning that we are looking for k-mers that are either unique to a gene or near unique to certain genes. So they are the informative k-mers. And then this inner product is distributive, right? So it doesn't matter if your summation is outside or inside, meaning that you can create a single vector from all the reads, assuming that if the k-mers are not originating from that gene, the read and gene dot product will give you 0 anyway. So you basically smoosh all your reads into a single vector and then do the dot product between that vector and the gene, and that gives you the proxy for that gene expression. So then how do we do this in homomorphic encryption? Now we define a very simple algorithm as a proxy for gene expression quantification. So we're going to use BFD. We will work in integer space. So inner product is nice because it's only multiplication and summation. So basically you encode your read vector into the coefficients of a polynomial, and then you encode your gene vector. And this vector, remember, the values are the frequencies of those k-mers showing up, how many times they show up, into another polynomial. And then if you multiply these two polynomials, the constant term of that multiplication is actually the inner product between these two vectors. So in basically in one shot, you can calculate the gene expression, the proxy of the gene expression, because you're smooshing all the reads into a single vector. So just one inner product is enough for that. So overall, what we are doing is basically, first, we are creating a k-mer table from our reference transcriptome, and then asking how many times we see those k-mers in our reads, and how many times we see those k-mers in our genes, creating two vectors with two polynomials, and then multiplying those polynomials all in homomorphic space and getting the encrypted gene expression results. So we can download these encrypted values and then decrypt it, and then that will be the. So this sounds too good to be true, right? Such a simple algorithm. And I kept telling my postdocs that, no, this is not going to work. This is too simple. You're not caring about, we get obsessed with the mismatches and all that stuff. But when you look at the results, using this as a proxy to gene expression, we basically compared pQuant to star and the Callisto, and then we saw that over 90%, the R squared is over 90%, and then we looked at star and Callisto, and that difference is within the expected range of difference that you would see between plain text. I wasn't still satisfied with this, so I asked them to do multiple samples and then calculate differential gene expression, do a downstream analysis to see how this affects the downstream analysis. So we did that, and then we compared the fold changes that we get from pQuant to star and Callisto, and then we looked at star and Callisto. It looks really good. But you can see that there are some issues here, and I'm guessing that these are. We are investigating this, but I'm guessing these are multi-map, perhaps reads that are, because we don't distinguish, all the reads are smooshed together, so you don't know which read is actually belongs to which gene that the read is originating. So there is a little bit of an effect to the downstream analysis, but it overall is pretty good for an encrypted algorithm. And then we looked at the runtime. So what we did first, we looked at per gene, because you can calculate each gene in parallel. They are independent from each other. So I think at most, and because all the reads are put together into a single vector, our runtime is not dependent on the size of the the number of the reads, basically. We get the same runtime for all number of reads. The maximum per gene was 3 minutes to calculate. But then what we did is we did this calculation where we the end-to-end time cost for all 60,000 genes in a given sample. So we use batches. So we calculate every 200 genes at once. And now when we did that, so it's around 3 to 4 hours for a single sample, independent of the number of reads. Oh wow, I went too fast, I guess. So as a conclusion, we have a method that can calculate gene expression pretty accurately in the encrypted space and fairly quickly. And this was recently published, so if you're interested in more details. And this was mostly done by my postdoc, Sengwan, who is a pure mathematician. He didn't know what a gene is when he came to my lab, so it was fun. Him and my other postdoc, Connor, who's a bioinformatician. And I remember when they first joined, I put them into a room, closed the door, and I said, we are not leaving this room until you guys teach each other. It didn't go well, so I took a different approach, but eventually we published this paper, and I'm very thankful for my funding and my lab, and happy to take any questions since we have time. Thank you.